So welcome to the third and final presentation in our plant responses topic. This lesson will cover gibberellins, abscisic acid, ethene and the commercial uses of plant hormones. Here's an overview of what this presentation will cover. So it will be the roles of hormones in the leaf loss in deciduous plants, seed germination and closing of stomata. We'll be having a look at the evidence of gibberellins in experiments and various practical investigations, and lastly the commercial uses of plant hormones. Starting with gibberellin. Gibberellin is a group of compounds, and the most common one is gibberellic acid. A bit like auxin, there isn't just one particular chemical that comes into this group. It is a combination of a different number of them. They all work in the same way though. What they do is they elongate the internodal regions of the stem. Now the nodes are the points where leaves or side shoots grow, and if we look at this diagram you can see that the internodal distance is much longer on the normal one, and the distance is reduced with the dwarf plant. Gibberellin is very important in breaking seed dormancy, and it does this by actually getting the food stores, the starch, broken down so that the plant can actually use it to grow. It works with auxin in this process and also in producing taller plants, and it can be involved in making fruits much larger. So if you expose gibberellin onto the plant, the fruit will stay on the plant for longer and the fruit will become bigger. So let's look at how gibberellins cause stems to become elongated. Gibberellic acid will increase the length of a stem. The picture you can see on the screen shows some dwarf plants, and some of the stems on the left have been treated with gibberellin and the ones on the right have not been given any gibberellin. Now I've mentioned that it increases the distance between the internodes, so that's the distance between where you would get side branches, and we've got the distance there on the left compared to that much shorter distance on the right. So you can see that the gibberellin has definitely lengthened those stems significantly, and it does this in a couple of ways. Firstly, it encourages the cells to divide, so you actually get more cells on those stems, and also every cell itself becomes longer. From a plant's point of view, producing gibberellin is actually a benefit to the plant because the plant will become taller, and if it's taller it's going to outcompete its rivals and get space and light and resources. So it's beneficial to a plant to have gibberellins. From a farmer's point of view, it might be beneficial to have less gibberellin because the plant, you don't necessarily want the plant to invest all its energy in getting really tall. When that happens, a lot of the energy of the plant goes into producing stems which aren't particularly productive, and also the plant then becomes tall and therefore can be pushed over if you get a windy day. So plants that are dwarf can actually be better from a cropping point of view than producing very tall plants. Scientists were keen to research how gibberellic acid and stem elongation were connected, and one of the things they discovered was that dwarf beans are dwarf because they lack the gene producing gibberellic acid. And Mendel did quite a lot of experiments using dwarf plants, and what he found was that dwarf plants tended to be recessive, and they were therefore recessive because they lacked the dominant allele which coded for gibberellic acid. So they simply didn't make this chemical and therefore didn't grow particularly tall. What we have been able to show is that plants with higher levels of gibberellic acid are taller, and if you administer gibberellic acid to dwarf plants they will go back to being tall plants. Gibberellic acid plays a very important role in breaking seed dormancy, and lots of plants produce seeds which don't germinate until the following spring. So the seeds are nearly all produced in the summer and autumn, and they are shed onto the ground and they remain there dormant until the spring. And so there has to be a way to actually break that dormancy. Now, in fact, the dormancy of the seed is caused by the presence of abscisic acid and other hormones. But to actually break that dormancy, you need gibberellic acid. So we're going to just have a quick look at the anatomy of this seed to start with. Within a seed, you've got a embryo plant, and that consists of a embryo root and an embryo shoot, and they will remain dormant until activated. We've also got a very large food store, and that's stored inside the region of the seed called the cotyledon. Now, if you've got one food store, that makes the seed a monocotyledonous plant, and if you've got two food stores, that makes it a dicotyledonous plant. Monocots tend to be things like grass, barley, cereals, that type of thing, and dicots tend to be pretty much everything else, but beans are a good example. So we get into the spring, and one of the first things that you get is a little bit more warmth. The soil warms up from the sun, and that also encourages uptake of water. Now, it is important to have both warmth and water, because there's always water around in the winter, but it's not particularly warm, so the seed is not encouraged to germinate. Now, also, the presence of abscisic acid might be making that seed resistant to germination anyway. But warmth and water will mobilize gibberellic acid, and gibberellic acid works on the DNA of the cell, so it activates genes in the nucleus of the cells of this seed, and that then creates messenger RNA, which then carries out protein synthesis and creates enzymes like amylase. Amylase will break down the starch, and the starch will then break down to release sugars, and those sugars will then be used by the embryo plant for respiration, and that respiration will then allow the embryo plant to start growing. We've already mentioned abscisic acid and its role in seed dormancy, and therefore it's working antagonistically to gibberellic acid and auxin. It's also got other roles. It's a stress hormone. When a plant is experiencing stress, for example, not having enough water or being too hot, abscisic acid will be released, and it helps it to cope with these conditions by, for example, closing its stomata, which reduces water loss. Or it could encourage the plant to start shedding leaves. So if we've got a very hot summer, then the plant might start to actually drop some of its leaves, and that will then also reduce water loss. Overall, it is very important in the role of abscission, which is the dropping of leaves and fruit from the plant, and that happens, of course, every autumn, and abscisic acid plays a very important role in that. So abscisic acid is very important in controlling stomatal closure. Drought will put the plant under stress. It will cause the plant to wilt, and that's also detected by leaves and possibly by the roots, and its response to that is to release abscisic acid. If we look at stomata in these diagrams, on the left we've got a normal stomata where the cells are full of water and the stomata are nice and open. The plant is experiencing stress, so it starts to release abscisic acid, and the effect of the abscisic acid is to cause calcium ions to move into the guard cells, and that then promotes the movement of potassium ions out of the cell, and those potassium ions take water out of the guard cells with it, and that then reduces the volume of these cells, so the cells become much less turgid and they collapse in on each other, and that causes the stomata to close. The next role of abscisic acid is in leaf abscission, which basically means the dropping of leaves, particularly in autumn. Now, deciduous plants produce large numbers of thin, fine leaves in the spring. The leaves are not particularly well protected. If you compare those type of leaves to the leaves of evergreens, evergreens like laurel are much thicker, tougher leaves, and therefore can withstand all kinds of damage and predators and diseases over long periods of time. Deciduous plants, on the other hand, produce these much thinner, more delicate leaves which are much more vulnerable to damage, predation and disease. And also, deciduous plants drop their leaves because by the time you get into the autumn, the light levels have fallen, and that means that plants are not photosynthesizing as much as they would do. So it makes much more sense to get rid of the leaves, cut your losses, and just become a bit more dormant over winter, where the light levels are low, the temperatures are lower, so all of these things will not be favourable for photosynthesis. And sometimes even the water availability is much less because, of course, if the ground is frozen, then you can't get the water. So I've covered the main advantages for a plant to do this. So reduces water loss to its leaf surfaces, avoids the damage of frost, avoids the fungal affections through damp, cold leaf surfaces, and that's basically because plants also have very limited ability to photosynthesize in winter anyway. There are 3 different plant hormones or chemicals involved in leaf fall. The first one is auxin. Now, auxin's job really is to prevent leaf fall, so it inhibits abscission. So when auxin levels are very high in the summer and the spring, the plant is dividing and producing lots of new leaves. We don't want them to fall off, so it actually has the opposite effect and makes sure that the leaves do not come off the plant. The next chemical is ethene. It's not really a hormone, it is a gas. It's the same material that you come across in your organic chemistry GCSE. And mature plants, particularly fruits, will start to produce ethene later in the summer, and this inhibits auxin production. It also activates genes which will help to weaken the cell walls, making it easier for the leaves to fall off. And the third hormone is abscisic acid. So when we look at the stages that actually lead to leaves falling off, the first thing that happens is as the leaves get older, the amount of auxin that they're producing starts to go down. They're not dividing as much as they did. That happens because it's under control of other hormones which can detect that the light levels are getting slightly less than they were at the beginning of the spring. Leaf is more sensitive to ethene production because the auxin levels now are dropping, and more ethene then is produced, and this inhibits the auxin production even more. The abscission layer begins to grow at the base of the leaf stalk. Now this is a layer which cuts off the vascular tissue and reduces the flow of materials in and out of the leaf. And eventually that abscission layer creates a complete seal over the vascular tissue, and it's made of a material called suberin, which is basically cork, and eventually this will be completely capped to prevent anything like fungi or bacteria or anything like that getting into the area where the leaf falls off. The abscission layer is then made up of these much thinner walled cells, and that's caused because enzymes have actually broken down a lot of the walls of these cells in this area, so they're now much thinner than they were, and this means that the leaf is now very vulnerable to being broken off. So a little bit of wind or a bit of disturbance and the leaves will start to fall. Just covers there a little bit more. It tells you about the cell wall with suberin, and that you get the scar, which is the capped off bit where the leaf fell off. This diagram just covers that in a little bit more detail. So what you can see there is, you can see this area is the abscission zone, and right in the middle you've got the vascular tissue which is being blocked off. so that now the materials cannot get in and out of the leaf, and eventually the leaf will start to wither, and eventually the cap will be so sealed that the leaf will just break off. This is a good moment to just remind ourselves about synergism and antagonism. So auxin is working antagonistically with ABA or abscisic acid during leaf abscission, so they're opposing each other. ABA and ethene, on the other hand, are working synergistically in leaf abscission, so they're both part of this process of causing leaves to fall. Gibberellins and abscisic acid are working antagonistically in seed dormancy. ABA will actually promote seed dormancy in the autumn, and quite often what happens is that the abscisic acid breaks down over the course of winter with particular types of seeds, and that allows gibberellins to become more effective. Gibberellins were going to work synergistically with auxin to promote cell division and growth, and that would obviously be particularly in the spring when the plants are starting to germinate and grow. So we're now going on to the commercial uses of auxin. The first one is if you spray plants which are producing fruit with auxin, it will prevent them from dropping their fruit. And this is a very useful way of controlling the dropping of fruit. For example, if you spray it onto your apple trees, the apples will continue to develop, and it means that also you don't have to keep going in and harvesting the fruit day after day, so you can actually control that harvesting process. If you spray plants with auxins, they can also produce seedless fruit. So if you spray it onto flowers, it will actually cause the fruit to be produced without fertilization, and that's particularly good if you don't want seeds in your fruit. Rooting hormones can be used by growers. So most rooting hormones contain auxin, and if you put it onto the end of a shoot, it will stimulate the shoot to produce more roots. I personally think that you don't need to do that because plants will do that anyway, but you can improve that process by using a rooting hormone if you want to. So here we've got some examples, and we also, the other one that auxins do is works as a weed killer, particularly on broadleaf plants. The way this works is, although it sounds counterintuitive to suggest that spraying an auxin which encourages growth will kill off weeds, what it does is you spray it onto the plants as they've done on the left here on this picture, and all of those plants will absorb the auxin and grow rapidly, and they grow so fast that they cannot sustain that growth and they eventually die. So that is basically how the auxin works as a weed killer. The next thing is ethene. Now a lot of fruits are harvested in a very unripe state, for example bananas, and that makes it much easier to pick the bananas and transport them without them being damaged. So when you then get them to their destination, you can expose them to ethene, and then they will ripen, and you can use that keeps your fruit in good condition. Not all fruits actually ripen this way. So for example, oranges don't particularly ripen very much after they've been picked because they don't produce much ethene. So gibberellins can be sprayed onto crops, and that will increase their growth. And if, for example, if we spray it onto a citrus tree, it encourages the oranges to stay on the tree for longer, and therefore they become bigger. It can also increase the yield of things like sugarcane. So growers will spray this on, and you get a bigger crop from your plant. Gibberellic acid is also sprayed onto barley seeds, and this then causes them to germinate, and you need this process in order to break down the starch to get the maltose, so that you can that can be used by the yeast to produce alcohol. So this is the process of malting. We haven't talked about cytokinins, not in this context, but they are a plant hormone, and you probably have remember hearing about them in terms of cytokinesis, and cytokinins will cause cells to divide. and they work synergistically normally with auxins and gibberellins. But they're very useful because if you spray them onto flowers, for example, or lettuce, it prevents the general breakdown and decay of those tissues, so you can keep flowers for longer. If you buy flowers from a supermarket, they do seem to last an eternity, and that is because they've been sprayed with these chemicals which maintain their lifespan. Cytokinin is also used in a process called tissue culture, which is something you're going to come across later on in the Year 13 course. So let's have a quick look at this Biology Olympiad question. Dormancy in seeds may result from the presence of which of the following? So I'll give you a moment to have a look and decide what you think the answer is. And hopefully you've got 1 and 3. So a high inhibitor content, which will be the abscisic acid, and an intact tester, which is the coating around the seed, preventing water uptake. Before we finish this presentation, I just want to spend just a few minutes thinking about some other plant responses. And one of the interesting ones is how plants know what time of the year it is. How do they know whether it is spring, summer or winter? And that will obviously affect its activity. So how does a plant know that we're going into autumn and I need to drop all my leaves? And it's spring, so I'm going to start germinating my seeds and producing new shoots. And the answer lies in a chemical called phytochrome. And phytochrome is a protein which can exist in 2 different forms, either as phytochrome red or phytochrome far red. And we've just got here the picture showing the molecule of phytochrome far red. So phytochrome red is the inactive form of this protein, and it exists when it's exposed to far red light, or basically dark conditions. So in dark conditions you only have far red light available, so that will cause this phytochrome to become P red. So phytochrome far red is the active form, and during normal daylight the phytochrome red will turn into phytochrome far red, and this will then activate this protein, and it will then activate other molecules and other enzymes, and it will also inactivate other things. So how does the phytochrome work? On the left here we have got the inactive form, phytochrome red, and that will be the form that would exist at night time. And on the right you can see that we've got the active form, which is the phytochrome far red. So during the summer the phytochrome will spend more of its time in the active form as far red, because the days are much longer, and so the active form is going to be promoting certain activities involved in growth. During the winter the inactive form is going to be more prevalent, because the days are much shorter and the night time is much longer, so the plant is therefore not going to do as much as it would do normally. So there has been some other experimental evidence though that shows that if you alternate red and far red light, then it doesn't matter how much time they spent in far red or red, it's just the last colour of light that it experiences which determines what it's going to do, which makes sense. So during the day it will be experiencing red light, so it will become phytochrome far red, and so it will remain in that format. And at night it will be experiencing far red light, that's the last light it's experienced, so it will remain in that format. But it wouldn't matter how much time it was exposed for as such, other than the fact that the last colour it saw will turn it into phytochrome red or phytochrome far red. But from a point of view of the way it works in real life, obviously during the summer the last light it sees during the day is going to be red light. So this just summarises what phytochrome far red can do. It could initiate some enzyme action. It could inhibit some enzymes. So some enzymes that are involved in reducing growth would be inhibited. It also regulates which plant flowers depending upon whether they are considered to be long day or short day plants. Now, long day plants are plants which recognises that we're in summer because we've got longer days and short nights, and they need to experience these long days in order to flower. Some plants, on the other hand, which live nearer the equator and don't experience long days at all, will require a critical amount of night and day in order to actually flower. So orchids, for example, could fall into that category. So again, this is a sort of detailed information which you could be given some experimental data on. Now that's all I need to talk about with the phytochrome, but I just wanted to mention it because I feel like it's one of those things that could come up even though it's literally a few lines in your textbook. Now this then brings me to the end of this presentation, and just to remind you again, this is a favourite topic of the exam board. I have seen questions on phytochrome in the specimen materials. I've seen questions on plant hormones in the majority of exam papers. So again, plenty of questions to look at. Please make sure you work through the questions that I've given you, and also be aware of the need to be able to apply investigation skills and data handling for this particular area of the course. That brings me to the end of this presentation. I hope you found it useful, and I hope you will be able to get on with completing all the rest of the tasks on the sheets. Thank you very much, everybody.